Welcome, everybody. We'll get started, though people are still coming in and beeping in, which is great. So we want to get started because we have a lot to tell you in this webinar. This is the NextGenMicrofluidics webinar on lab-on-a-chip technologies for biological monitoring. This is the first in a series of final dissemination webinars we have for this project, which is ending in March. So we're happy that you're here to learn about what we've been up to. The agenda for today looks pretty straightforward. So we have a short overview of the project by our coordinator, Martin Smolka of UNM Research. Then we have two presentations from two of our demonstration cases. The first one is from Ayerdi Izquierdo from TECNALIA, and then the second from her colleague, Verónica Mora Sanz, also from TECNALIA in San Sebastian, Spain. And I'm Caitlin Ahern from BioNanoNet. We're a partner in NGM project doing communication dissemination, as well as project management. So we're happy to have you here. At the end of each presentation, we'll have time for Q&A, and also at the very end if you need some time to think of your questions. You can write questions anytime in the Q&A function. You can also use the chat if you need anything. And we look forward to hearing your questions and feedback at the end. So Martin, I'll hand over to you for an overview of the project.
So hello, everybody. First of all, in the EU project NextGenMicrofluidics, we identified a rather big challenge in the development of microfluidic devices. So first of all, as you know, development of microfluidic devices is complicated, and typically you need to combine the solutions from different providers. So you need different entities in your chip, and you need to contact different players for your development. And since this microfluidic market is still rather new, typically the customer needs to choose the partners and need to verify that the different technologies match to each other. This challenge we are addressing with our approach, and the approach is that with our 17 million EU project, we first of all combined 21 solution providers, which cover the entire value chain for microfluidic device development and production. And we founded our own startup. So through the project, we founded the Microfluidics Innovation Hub, in short, the MIH. And now you can approach the MIH as a single entry point to our services. So if you want to get a combined service offer where we all contribute to the solution of your problem, of your development application, then you get a combined offer through the Microfluidics Innovation Hub. And yeah, through the project, we got a nice chance, first of all, to verify our technology. So we had several demonstration cases, and I'm very happy that we have the chance to hear more about two of them today, by Verónica and by Anne. And part of the project we also got through an open call program. So we went out to the world and asked customers what issues they have in microfluidic device development and how we can solve their problems in development. And all in all, so we worked for 22 applications. So we have already a broad experience demonstrated, and yeah, we have shown our capabilities and all these advancement of microfluidic devices. So all in all, with the Microfluidics Innovation Hub, we do now contribute to the microfluidics revolution in a leading role. So starting with a service portfolio for manufacturing of consumables, of the chips, the cartridges which you need in microfluidics. We integrate sensors and biosensors with our specialization in chemical field and biochemistry, and also in biological processes through the broad experience of our 21 service providers. And yeah, with all that, we work on lab-on-a-chip applications, so point of care and in vitro diagnostics, and also in on-chip cell cultivation, and so in so-called organ-on-a-chip applications. So just contact us for your development of microfluidic devices. just by getting in contact to the Microfluidics Innovation Hub. And yeah, just a short overview again of our partners. And with this overview, I'm happy to hand over to the presentation of our applications.
Great, thank you so much, Martin. And we'll move on to our first demonstration case application with Anna.
Thank you everyone for joining. Hello, I'm Anna Ayerdi. I hope you can hear me now. Yes? Okay.
Yes.
Today I will present the development of a microfluidic chip for neuron cell culture and axon outgrowth monitoring within this NextGenMicrofluidics European H2020 project. As Martin has just explained, and as you can see from the logos here underneath the title, top research centers and industry leaders all over Europe have come together within this project for this demonstration case. Okay. Advanced neuronal cell culture techniques have enormous potential to aid the discovery of new drugs for pathologies in the nervous system, a goal that has been historically quite challenging. For studying the in vitro formation and function of neuronal networks in drug screening assays, axon outgrowth should be ideally monitored. However, there's no polymer-based chip commercially available so far that allows this monitoring in vitro. To this aim, the NextGenMicrofluidic Consortium has designed and scaled up the manufacturing of a neuron cell culture chip that would allow the cell culturing and the axon outgrowth monitoring at the same time. Among the challenges that the consortium has faced throughout the project, some of them has been that the design should allow the culture, the growth, and the monitoring of these neuronal projections during the assay. This development should be roll-to-roll printable, non-toxic, and should include cell adhesive and micropattern surfaces. And it should also integrate stimulating elements or electrodes in the chip. So the workflow that I am going to present in today's webinar will start from the chip design. I will show you also the microfluidic simulations that led us to validate this chip design, the material development and upscaling of the chips, the development of the stimulating device, and finally some functional validations. As for the chip design, it was agreed since the beginning that the chip should consist of separated chambers for cell culture, and those two chambers should be connected by microtunnels with few micrometers in size. This chip should also be compatible with a 96 well plate format, which is the regular consumable that we use in cell culture labs. And as neurites will be projected throughout those microtunnels, and those projections should be recorded by fluorescence microscopy, the substrates that we will need to use, there's the materials, should be compatible with this microscopy technique. Hence, the chip was envisioned as the merger of three layers. The bottom layer will consist of screen-printed electrodes that will be bonded to the middle layer. This middle layer will be containing the microstructures, and these both layers will be afterwards interfaced within a 96 well plate. Once the design was set, microfluidic simulations were performed to check the air bubble formation and to determine the molecule diffusion throughout the microtunnels. This simulation determined that the diffusion of BSA, which was used as a protein model, takes up to 14 minutes until the concentration is homogeneous throughout the chamber. Most importantly, 75% of the BSA is diffused after 6 minutes, which in all accomplishes the goal of nutrient delivery in a very reasonable time for these types of consumables. As for the material development, first a screening of which material will be used for electrode printing was done. This material will not only allow the culture of neuron cells for up to 11-15 days, but also should allow the screen printing of electrodes on it. As a result of this screening, COP was chosen as the material. And electrodes were printed on both sides, with leads and insulation printed on the bottom side of the substrate, and the conductive pads were printed on the top side. Connections between the two sides of the substrate was obtained by bias, cut by laser before printing, and PSS was used for printing the conductive electrode pads. As for the manufacturing of the middle layer, two strategies that were compatible with roll-to-roll manufacturing were followed: UV nil imprint and extrusion coating. For UV nil, in the UV nil strategy, a polymer master was fabricated, from which a PDMS replica working stamp was obtained. And here we can see an in situ monitoring of how this batch replication looks like within the roll-to-roll production line. For the extrusion coating manufacturing of these structures, the medium layer, a similar strategy was followed. In the left side of the slide, you can see the SME micrograph as a quality control of the stamp that we were using for this replication strategy. And here we can see also a small video of how the batch replication process looks like in real time. Yes, but once both layers were manufactured, then the upscaling of the tip was addressed. Bonding the microstructure layer to the bottom layer has proven to be a very challenging step. The requirements that this first bonding should meet were that they should be roll-to-roll compatible, the bonding method should be compatible with a later implementation in a roll-to-roll production line, biocompatibility, because the chosen method should not affect the culture cells in a detrimental way afterwards, and the preservation of the microstructures as critical features of this chip design. Different bonding strategies then have been used depending on the nature of the bottom layer and the source of the microstructure layer. But in any case, here we can see how the final tip look like, and how the microstructure side of the design, maybe I can use the pointer, I don't know, how well aligned is with a 96 well. For these electrode-based chips, a multi-channel electrical stimulation platform has also been developed within the project. It has been referenced as Steam4Cell, and it operates in two modes: constant or pulse current. The output current values range from 0.001 to 10 milliamperes, supporting pulse frequencies from 10 Hz to 400, with a minimal pulse width of 550 microseconds, allowing us to perform a broad range of stimulating protocols in order to get the successful one for each cell line that we have worked with. This Steam4Cell platform is controlled by an Android application that we have also developed within the project, and that allows to set individually for each of the stimulating chambers that are contained within the microchip, the amplitude to be individually set for each of those stimulating chambers, whereas the pulse width and the frequency are set for all the paths altogether. So the in vitro validation, the functional validations that I'm going to show from now on has followed the same protocol. All the developed chips have been in vitro validated using cell culture, using cell cultures. For that, these assays have started with plasma treatment and a biocoating. Then cells were seeded inside the chips. Media was changed every other day, and the monitoring has done using inverted microscopy. So first of all, I'm going to show you the results that we have obtained without applying any electrical stimulation. These are only with chips without applying electrical stimulation. And here what we can see is the result that we have obtained with several cell lines. In the left side, we have dorsal root ganglion cells that were firstly isolated from rat embryos and seeded them on the chips after coating them with polylysine in this case. After 6 days in culture, primary cells were stained with calcine. That's why you can see green all over the micrographs. And what we can see is that cells are well spread all over the macrochannels. They are starting to project neurites throughout the microchannels, and they remain functional over time. As for the picture in the right side, what we can see, this is another cell line. This is an immortalized cell line, and this time it was stained with phalloidine in red. We also obtained that cells are well spread all over the macrochannel and starting to project neurites throughout the microchannels. This chip was also used for developing an in vitro neurodegenerative model. The interaction of Schwann cells with neurites of DRGs plays a crucial role in axonal myelination in vivo, suggesting or making this model a very interesting tool to study or replicate in vitro this vital process. So to develop this assay, DRGs were seeded on one side of the chip, and after 4 days in culture, and as the neurites extended through the microchannels that can be seen slightly here in the upper side of the picture, as the neurites extended through the microchannels, then Schwann cells were seeded in the other part of the chip and allowed to adhere to the axons. So in the micrograph, we can see several colors. Red colors are Schwann cells. And in these white arrows, what we can see is that these Schwann cells can indeed adhere to the neurites of the DRGs that were seeded on the other side of the chip, making this co-culture a very representative tool for studying axon myelination in vitro, as it replicates this interaction that happens in vivo. As for the functional validations that we did using the Stim4Cell device, that means applying electrical stimuli on the developed chips, several cell lines have been used for this in vitro validation. First, using these NE4C cells, which is an immortalized cell line, and after applying different protocols, stimulating protocols, it was seen that when 5 milliamperes was used, more cells were counted within the stimulating chamber when compared to the non-stimulated chambers. In fact, when these pictures were analyzed under image day, we also obtained not only that more cells were located when stimulated with 5 milliamperes, but also the cell body length was higher when compared to the cells non-stimulated. Electrostimulation was also studied using DRGs, and with these cells, different endpoints were analyzed. First, BDNF release was quantified. Neurotropins play a crucial role in the brain, as they are essential for the survival, growth, and differentiation of nerve cells. In natural conditions, neurotropins are released in response to electrical stimuli in the brain, making them, or the quantification of these neurotrophins, suitable candidates to assess the device functionality in activating both neural and glial cells. So when DRGs were stimulated using different protocols, what we obtained was that these cells were able to produce more BDNF in an amperage-dependent manner after electrical stimuli when compared to the non-stimulated cells. Morphological changes in axonal growth using these cells were also recorded, and what we could obtain is in the micrographs of the center of the slide is that when compared to the non-stimulated cells, neurites have a different pattern, as when stimulated they are more aligned rather than in the non-stimulated where they look like all over spread and without any alignment. And also, when primary parameters analysis were performed, what was quantified is that DRGs after electrical stimuli appear to have a higher neurite number. These neurites were longer in length, and also the extent of branching was increased in comparison with the non-stimulated control. And finally, these chips were also validated using Schwann cells. When activated, these Schwann cells also release an NGF factor, which in turn activates other neuronal cells. And a significant increase in NGF release was obtained when Schwann cells were stimulated using different in a current-dependent setup. So all in all, the microfluidic chip that has been able to that the whole consortium has been able to manufacture allows the correct growth of both primary and immortalized neuronal cell lines, allowing significant growth of neurites and allowing also the recording or the monitoring of this axon outgrowth. Besides controlling both the amperates and the stimulus duration with the multi-stimuli platform that we have developed within the project, we can obtain a robust response in the cells. So very briefly, the following scheme shows the manufacturing process that has been followed and each partner involved in each step. I can assure that the complexity behind the manufacturing of this chip and its upscaling could not have been addressed without the contribution of all the partners listed in this slide. And with that, I would like to thank you all for your attention. And of course, if you have any question, I will more than welcome to address it. Thank you. Yes.
Thank you very much, Anna, for your presentation. If we have any urgent questions right now, we can take them. And otherwise, we might save questions for the end in the interest of time. But if anybody has a quick question now, you can write them in the chat and then I can unmute you if you want to ask them yourself. You are all automatically muted. But if you write them in the Q&A. And then otherwise, we will move on from now to our second presentation from Verónica. And then please be thinking of your questions for the end. You can write them in the chat at any time, and then we'll come back to them after Verónica's presentation.
Good afternoon, everyone, and thank you for joining our seminar. I'm Verónica Mora Sanz, a researcher in the biomaterials area of the health unit in TECNALIA, and today I'm going to present a demo case developing the NextGenMicrofluidic project about enzyme activity monitoring in industrial solid-state fermentation processes based on colorimetric lab-on-a-chip compatible with roll-to-roll fabrication. This project was made thanks to the collaboration all of these research centers and industries in Europe, each one contributing with their expertise. We work with a German company, Naturstofftechnik, that produce a biodetergent. And they wanted to control the concentration of amylase and cellulase that are the most abundant laundry enzymes in their composition. And they wanted to control the enzymatic activity in situ and at line. But the current challenges that we have when we want to monitor the concentration of enzymes in solid state fermentation process is that we need to have specialized equipment and also trained personnel. And sometimes the methods are labor extensive and costly. And this makes that these methods are not suitable for real-time monitoring and for real-time making decisions. So lab-on-a-chip technologies emerge like a promising solution because these devices integrated several laboratory functions into only a single chip. So here we propose the design and the scaling up of a colorimetric lab-on-a-chip compatible with roll-to-roll extrusion coating fabrication. Some advantages of using a lab-on-a-chip are the miniaturization, that they are very fast and efficient. We need a very low amount of sample and reagents. They are portable, cost-effective, and sometimes the sensitive is enhanced. During the project, the complete value chain has been fulfilled, starting from the chip design and computer-based optimization, and also the scaling up of the chip production, employing roll-to-roll extrusion coating. We also make the integration of all the reagents into the lab-on-a-chip, and we also develop a customized portable device adapted to the chip geometry. We set up a method for the extraction of the enzymes from the samples of Naturstoff, and we finally also made the functional validation of the chip and the reader with these real samples. So here we can see a foil with several units of the chips produced by roll-to-roll extrusion coating. Here we can see a single unit of the chip that is composed by an inlet, a detection area, and also a capillary pump. We have to introduce the enzymatic substrate here in the reaction chamber on the chip, and to apply an hydrophilic reagent in the capillary pump. And then we have to close this chip, and for this we use a pressure sensitive adhesive tape. We close the chip, and then it is ready to be used. The chip works on the principle of capillary-driven flow microfluidics. The user adds the sample containing the enzymes by the inlet, and the chip starts to self-fill. The liquid arrives to this detection area where the reagents are located and where the reaction takes place. And then the liquid continue the flow until it arrives to this capillary pump that removes the liquid exit and isolates the reaction chamber. Well, the chip design was improved by computational fluid dynamics simulations. To know the pressure at which the meniscus is raised was a very important parameter. And for this, here we can see a model of the inlet. Here we can see that this meniscus is formed at a pressure of minus 160 pascals. And then when we achieve a suction pressure of minus 270 pascal, the meniscus is broken and the liquid starts to flow through the chip. We test several capillary designs to see which one provides a good pumping strength, and we achieve the best results with the micropillar design. Here we can see an image of this microstructure. There we can see the micropillars of 100 micrometers. Here we can see how the chip works. We introduce the liquid by the inlet. The liquid arrives to the detection chamber. Here now we are going to see how the meniscus is formed and then is broken. As we can see, the liquid is now has arrived to the capillary pump, and then we can see how the reaction chamber is isolated. So the scaling up of the chip production is performed by roll-to-roll extrusion coating. That is a continuous process that is used for flexible materials supplied in rolls. The substrate unwinds from our rolls, passed through an extruder that melt the plastic, and then the molten film is coated onto the substrate via rollers. And after cooling and solidifying, again, the coated substrate is rebound into a new roll. We decide to employ this material, cyclic olefin copolymer, COC, because it met all the required criteria that we needed. It is flexible, required for roll-to-roll, thermoplastic for the extrusion coating, and also due to these optical properties, because we need that the chip is transparent to be able to measure this colorimetric reaction. The advantages of using the roll-to-roll manufacturing are in terms of efficiency and productivity due to the high speed and scalability, the cost reductions due to the material use and energy efficiency, also because we have a lot of flexibility in applications, the consistency and quality of the processing, and also because we could use it, for example, for rapid prototyping to test several types of chips or microfluidic pumps. Also due to the environmental impact, because it reduces waste, and due to the lower energy consumption. Well, as I explained before, we are going to be measuring two enzymatic reactions, and for this we need to employ some enzymatic substrates. To simplify the handling and to reduce the user interaction, we introduce these reagents into the reaction chamber of the chip, and we employ different methods. Here, for amylase, we need to employ two different reagents, an auxiliary enzyme, alpha-glucosidase, and the chromogenic substrate, PMP-G7. By the action of both enzymes, this compound, PMP, is released, and we can measure the absorbance at 405 nanometers. So the integration of the reagents in this case is making by manually drop casting directly in the reaction chamber of the chip. This drop is immediately freeze, and then the solvent is removed by lyophilization. By using this method, the shelf life of the chip is of 1 month at 4 degrees under inert atmosphere. Here we can see an image of the chip with the reagents in the reaction chamber. Then we add a solution containing amylase, and we can see how the reagents are solved, and the liquid turns yellow due to the formation of PMP. In the case of cellulase, we just need one substrate, resolving cellobioside. By the action of cellulase, the dischromogenate compound resolving is released, and it has absorbance at 570 nanometers. In this case, the integration of the reagents was made by spotting by piezoelectric deposition directly in the reaction chamber of the chip, as we can see in the video. In this case, we are spotting a very low quantity of volume in the range of the picoliters. This is why the solvent is immediately evaporated. This technique is compatible with roll-to-roll manufacturing. And in this case, again, the shelf life of the chip is of 1 month at 4 degrees under inert atmosphere. So, as I said before, during the project, we developed a customized colorimetric reader with the detection chamber adapted to the chip design. This reader is able to measure absorbance at 2 wavelengths, at 405 nanometers for amylase and at 570 nanometers for cellulase. It also incorporates heating elements and Bluetooth connection, allowing it to be controlled by a smartphone employing an Android application. Here we can see the flowchart of how we use the portable reader with the chip. We have the chip, we introduce the sample containing enzyme, and when the chip is full of liquid, we introduce it in the detection chamber, and then we start the kinetic measurement, controlling it with the smartphone. Naturstoff technique produced the laundry enzymes employing solid-state fermentation processes, where the microorganisms grow on solid materials without free-flowing water. This method is efficient and also cost-effective, enhancing the production of enzymes by mimicking the natural habit of many fungi. So here we can see a very simple procedure that we set up to extract the enzymes from the samples in the fermentation plant. We first weigh the samples employing a digital spoon or an analytical balance. Then we add the reaction buffer, and we perform a mechanical extraction using glass microbeads and by shaking with a vortex. And finally, we eliminate the solid residues by filtration. Finally, we perform the functional validation of the chip and also the portable reader employing these real samples. The samples were taken at different time points of the solid-state fermentation process during the growth phase of the microorganisms. So we expect to have increasing enzymatic activity in these samples. And of course, the measurements were carried out with the miniaturized colorimetric reader. The reader makes kinetic measurements. It measures absorbance in function of the time. We can see both results for amylase and cellulase. Here we can see that as the fermentation process progresses, the slope of these kinetic measurements also are higher for amylase and also for cellulase. Then we transform these kinetic measurements into enzymatic activity, and we can see that the samples show increasing concentration of both enzymes, indicating that as the fermentation process progresses, the amount of generated enzymes is higher. These measurements were quite fast because they only take 5 to 10 minutes. That is much faster than conventional colorimetric assays, and of course much faster than sending the samples to external laboratory. We can say that this lab-on-a-chip technology is effective for the quantification of amylase and cellulase activity and enzyme-containing ferments and works for a quick analysis in situ at a line in industrial solid-state fermentation plant. So the conclusions of this work is that during the NextGenMicrofluidic project, we have been able to produce a microfluidic chip produced by roll-to-roll extrusion coating for the detection of amylase and cellulase in one step. We have also tested two methods to integrate the reagents in the chip. On the one hand, drop casting and freeze drying, and on the other hand, spotting followed by air drying. It was possible to measure the enzymatic activity of both enzymes by following the increase of absorbance in function of the time with a customized miniaturized colorimetric reader also developed during the project. We also set up a fast and simple protocol to extract enzymes from the sample in the production line. And the performance of this Lavona chip was tested with enzyme-containing samples from Naturstoff, and a correlation between the sample time point and the enzymatic activity was achieved. Finally, the end-user side, Naturstoff, concluded that the Lavona chip technology works for a quick analysis of enzymatic activity of amylase and cellulase in enzyme-containing ferments and formulation thereof. So thank you all for your questions, and I'm very happy to answer any question you have.
Hey, thank you so much, Verónica Mora Sanz from TECNALIA, for your presentation. You're unmuted, so go ahead.
Yeah, hello. Well, thank you for the presentations. My questions are about the reagent integration strategies that you follow. I would like to know why you selected two different processes and how did you cap the chip to avoid damaging the reagents.
Okay, we test two different methods. We first started with the spotting. We prefer this method because it's compatible with roll-to-roll processing. However, when we try to spot the glucosidase, in the case of amylase, we didn't obtain good results because this enzyme got damaged during the piezoelectric deposition, and this is why for the introduction of the amylase reagents, we selected this other technique. And the other question was about the adhesive tape, how we do it to put it.
Yeah,
the cutting with it's a laser cut. Sorry.
No,
this was your question.
Yeah, the capping, like the closing the device after you deposited the reagents, especially with lyophilization.
We do it with manually.
Okay.
Yes, we don't have any special matching, but the chip is not a flat chip. It has microstructures, so if you put the adhesive tape, is like that you are not touching what is in the reaction chamber of the chip, or spotting, or with freeze drying. It's more or less flat.
Okay.
I would have a question in the meantime for Anna. I'll jump in here then. Can you tell us about the dimensions of the microchannels? How did you choose the ones that you did?
Well, yes, that's a good question because these microchannels were 3D, right? So what we did was during the design process, we were able to print microstructures with different lengths. We tried 150 microns, 450 microns, and 900 microns of length. Whereas the width or the distance among each microchannel was always set at 5 microns, and the depth of the microstructure was 10 microns. And the in vitro validation assays that we did with several neurons, because it's true that cell culture are usually tricky, but neuronal cell cultures are trickier. So at the end, yeah, we got the 900 micron length structures to be the ones within the chip.
Okay, thank you. And did you struggle with the bonding of the chips? And
well, yeah, that was a very,
can you tell us about it?
yeah, we struggled a lot. I think or imagine that after the bonding, we need to introduce cells. They do not like usually any trace or leaf of any chemical residue. So I think that we tried up to 6 different or 7 different type of bondings, depending on the source of the microstructure layer. And at the end, yeah, we were able to come with a roll-to-roll uvinyl based bonding technique when the microstructure was bonded to a plane surface, but when electrodes were on the bottom layer, a transfer adhesive tape was the one chosen for the bonding method because we could apply some pressure and both layers were stick together without many leakage observed and without toxicity related to the cells. Yes.
Okay, thanks. Veronica, do you think that your chip could be used to detect other enzymes?
Yes, yes, I think so. This is one of the main advantages of this chip design, that it could be used in other industry that employ enzymes, because we just need that this enzyme can be detected by a enzymatic reaction that release a chromogenic substrate. If, for example, we employ any enzymatic substrate that relates something different that didn't absorb at 405 nanometers or of 170 nanometers, we just need to change the LED of the portable reader, and it could be adapted to other substrate and other enzymatic reactions. Yes.
Okay, sounds great. If there are no other questions from the audience, give another second. Then we hope that you use the links to follow the project. You can also find videos about both of these demo cases with some more information. And if you want to see a little bit more about how they really look like, you can find them on our website. Follow us on LinkedIn for all the rest of the results coming from the project. And we'll be announcing the final 2 webinars, dissemination webinars in the new year. I'd really like to thank our speakers today from TECNALIA and UNM Research for sharing your results with us, and for all of our participants for coming. We hope you enjoyed it and keep in touch. Thanks a lot.